If there's no one, we can switch to the next session, so which is about a proposal, an answer to the PEPR FairCarboN goal, which maybe future collaboration between BIAM and LSCE. So it's Jean is starting. Okay.
Can you see my screen?
Not full screen.
Good. So, as you said, it's for the moment at the stage of the grant proposal, so it's a little bit embarrassing to speak about it before we've actually started doing anything. But it's basically a project aimed at trying to reduce the nitrogen inputs in agriculture, trying to use low chlorophyll plants. To give you a little bit of background on the call, well, it's not a call for scientists really. It's more a call for superheroes, I think, because to address the main objectives, you have to be able to integrate science from the molecular level, from the mechanistic molecular levels at the level of the chloroplast and the cell, up to increasingly integrated systems like plants in the field and the environment in response to changes in temperature, response to drought, CO2 availability, limitation of nutrients, these kind of things. So it's a really high impact projects, and as scientists, we have to recognize how hard it is to join forces to be able to address these extremely ambitious aims. So when we were thinking about these projects, we were actually also addressing questions of possible interactions between our respective institutes, because at BIAM, for example, can I use, yes, maybe this. Well, as plant biologists, we use the little plant Arabidopsis as a model species for our plant science. And we have to recognize that this tiny little plant is not really relevant to feed the global models like the land surface model Orchidea. At the moment, assimilation data used in Orchidea come from satellite data and flux towers data, eddy covariance data. It's drawn here, for example, towers in forests that measure the exchange of CO2 between the atmosphere and the ecosystem. So at BIAM, it's been shown already, we have growth cabinets to grow plants. They are airtight, so we can label plants with stable isotopes and follow CO2 assimilation or nitrogen uptake. And we can also measure the isotopic discrimination between C13 and C12 in plants. So this really, this is the big question. How can we talk to each other? How can we interact with each other? And it's not only a matter of scale. It's not because Arabidopsis is only a small plant and that the forest is much bigger. It's also because we have to find common models. So this, two examples, two pictures from two examples. That's the Tour à Flux, what's the eddy covariance tower at the Oak Observatory next to Cadarache. And so that's the oak forest and the CNRS lab, which is there. And that's the wheat field for experimentation, also next to us at Cadarache. And they actually correspond to two different models, which are used for orchidea. So this one would be the plant functional type 6, that would be for an oak forest, and the plant functional type 12 would be for C3 crops. That's typically a wheat field. So if we want to interact with each other, we have to, I'd say, we have to choose either one or the other of these models. Or there's another possibility, and people are thinking about that. It's to work on acclimation and regulation of photosynthesis, and about the hypothesis there might be an optimum for photosynthesis. So Nicolas is going to talk about that right after me. And also about the possibility of translating our knowledge from model species to crop species. And that's now possible using gene editing. So it's just about time that we think about the best ways to interact with each other. When I was discussing with Fabienne Maignan, it was about 5 years ago, I guess, about the possibility of using experimentation in the field, in this Arvelis field. I asked her, can you actually see this wheat field from your computer at LSCE? She said, yes, more or less. So using the data from the Sentinel-2 low orbiting satellite, she sent me 2 images back. So the first one for the month of May, and the second one is for the month of July. So that's after harvest. And there you can recognize the 2 functional plant types, the bare soil, no plants, and the wheat at the growing season. But you can barely distinguish the different experimental plots. So the spatial resolution is just about there, so we can make a link between measurements in the field and observation from satellites. So maybe I should skip this. So when we started to talk to each other and to find a way to integrate knowledge from the molecular level up to increasingly systems to the ecosystem, we thought we have to choose an observable which is really clear, a molecule that we can trace from the molecular level to the ecosystem level. And we thought, maybe chlorophyll is not so bad, because chlorophyll you can measure in the lab, and chlorophyll you can also measure from space. So we put together a group of different partners. So that's the BIAM partner, that's an INRAE partner, Fabien Chardon from INRAE Versailles. He's a specialist of nitrogen uptake and remobilization in crops. There's the partner Arvelis for the field trials, and there's the partner, the LSCE partner, and this is Nicolas Viovy, Fabienne Maignan, and Nicolas Vuichard, who is going to speak just after me. And our aim is to understand what changes in the chlorophyll content, how they impact carbon assimilation, nitrogen assimilation, and using measurements in the field and models, agronomic models, and then global models, how it may change the carbon and nitrogen cycle. So if I have time, actually the chlorophyll biosynthesis pathway is highly regulated. It's a bit boring to look at that. Let's just have a look at the last step. It's the reaction catalyzed by the CAO enzyme. Its CAO is for chlorophyllide A oxygenase. It basically regulates the ratio between chlorophyll A and chlorophyll B. And we have various cultivars affected for this regulation. So chlorophyll is embedded in large transmembrane protein complexes, so decreasing the chlorophyll content also decreases the protein content in the chloroplast. And this should have a major impact on the nitrogen content of leaves. And this is a picture of the different body varieties we have affected for the chlorophyll content. As you can see by eye, the pigmentation is very much different. And the amount of chlorophyll per leaf area varies in a large range. And this is our working hypothesis. By decreasing the chlorophyll content in the leaf, we are going to decrease the nitrogen from the soil, and we may also affect the grain filling. The kind of hypothesis we...
David, you're talking.
And now I think I should let Nicolas speak for the next bit of the research project. Are you ready, Nicolas?
Tout ça, bien.
Ouais, je crois que je l'ai fermé avant cette partie. So on my side, I will more focus on what we plan to do within this proposal at LSCE. So it will be complementary to what has shown Jean. So, and just not to repeat what has shown Philippe a few minutes ago, but just to highlight in our modeling scheme, I mean, we aim to model jointly energy, water, and carbon and nitrogen cycles, and that in this respect, there are, I mean, key processes that also have been highlighted in a former presentation, especially regarding water and carbon cycle, which is related to stomatal conductances. And that, just to highlight that we don't want to just look at or invest our work on the photosynthesis per se, but also, I mean, in the coupling, we have this importance of coupling and to account for also water limitation and the coupling through stomatal conductance. And so in this respect, the way we model, so I will avoid a bit as a highlight Philippe also the wording process-based, because I think it would be a shame in front of BIAM, but I mean, let's say it's more like a phenomenological model, so we tend to represent behavior or phenomena that we know acting on the ecosystem. And so typically, regarding photosynthesis, so keeping in mind that, okay, we do assimilate, I mean, we do model assimilation, so that we name here A, with two limiting factors, one coming from the Rubisco activity, so Rubisco limited rate of CO2, AC, and one that is related to the light limitation through the electron transport and the limitation of the RUPP regeneration. We have to account also for the gradient of CO2 at the atmosphere interface, so CI and CA, and the stomatal conductance, and all these equations, or processes or variables are connected together, and that we aim to solve jointly this set of three equations to get the free value of A, CI, and GS at each time step of our modeling framework. And so if I focus more on the A variable, so the assimilation, so that will be the key focus of our involvement in the proposal. So as I said, so we have two limiting factors, one coming from the Rubisco and the other one from the electron transport. And so we have two key parameters for these two respective equations. So the VC max for the Rubisco limited rate, CO2 assimilation, and the other one is variable J that corresponds to the transport of electrons, and that is on its side limited by this maximal value that we call J max. And so the way we parameterize these two parameters, so VC max and J max, are a bit, let's say, empirical or observation-based. So typically we have colleagues in the community of also trait community that tend to collect observation of these different parameters for different type of ecosystem, and it shows that, so, at the ecosystem level, typically we may assume that, okay, VC max is a linear function of the leaf nitrogen content, and so we have a kind of function where we have the VC max that is a function of the leaf nitrogen content, so multiplied by a function, a parameter that is the nitrogen use efficiency that might be PFT dependent. So at the level of the plant functional type we work with, this LUE may differ from one PFT to another. And also from the same group of colleagues from MPI Jena in Germany, it shows that at the, let's say, PFT level, we may assume that we have also a linear relationship between Jmax and Vcmax with a kind of stoichiometry of 2. Also, if we account for acclimation factor that may change according to the temperature, this ratio, but quite slightly, and anyway, it's quite a rigid relationship. So anyway, we assume similar co-affectation of nitrogen, indeed, towards, let's say, Vcmax, or what is behind Rubisco, or Jmax, and what is behind in terms of nitrogen. In parallel, and it's part also that we aim to use and to further develop in the proposal. It has been shown also by Philippe, so Fabienne and her colleagues are mostly involved in fluorescence and how we can benefit of this measurement and this modeling into Orchidea to better constrain the photosynthesis. And so typically, they develop, and that's what we want to update a bit in the new, reference version of the Orchidea model that we aim to work with. They develop an extended model of the model we had so far, but that do account for the different fates of absorbed light, and not only accounting for the one related to photosynthesis and the transformation towards chemistry and the formation of sugar, but so typically accounting for NPQ and fluorescence. So this model is the one of Johnson and Berry that you start in, and you have already a version into Orchidea that we aim to work with and to improve within the framework of the project, if funded. So just to summarize a bit the idea, so far we have a kind of fixed relationship between leaf nitrogen content, VCmax and Gmax parameter. A first attempt of the project for us will be to tend to revisit, to reparameterize this VCmax Gmax relationship in such a way that we can simulate the different varieties of barley that will be monitored at plant and ecosystem scale in the different work package of the proposal, and to aim to simulate the observed change in photosynthesis activity. So this is the first attempt. We would like also to explicitly account for the chlorophyll content, since so far, I mean, actually, we don't have an explicit formulation of chlorophyll within our modeling scheme. And the question is more how we could improve our modeling, accounting for this explicit representation of chlorophyll content into RTD. And last, it's the use of the fluorescence. And the question here is more like how we can aim to use fluorescence and fluorescence modeling to better constrain our modeling of photosynthesis. So that's mostly the space of involvement we would like to perform in the project, and in where we are quite really interested in this opportunity. I mean, for us, if you start to see the gap between what you can do at BIAM and what we can do in terms of modeling with RTD at LSCE, I mean, the gap is quite enormous. And so, I mean, such a project, it's also an opportunity to better know what we can do from both sides and how we can take advantage and also learn, I mean, just for our own interest. Yeah. So that's all on my side.
Nicolas. We have time for one question.
When you take into account nitrogen, you take total nitrogen, or how do you distinguish mineral nitrogen, or which is stored in the vacuole, and the one which is in the organic molecules?
No, so what I mean, in terms of form, where it's typically a single pool of nitrogen, we do account for a location of, I mean, where the nitrogen is. So, well, remember, okay, we don't have a leaf model, but we have let's say, a plant model or at ecosystem level. So typically, where we need to locate a minima where the nitrogen is stored, or for which processes or purpose. So typically, well, it's maybe something Philippe showed, but okay, we have simple pools of biomass indeed, root, stem, leaves, and so these pools are used also to locate carbon and nitrogen. So, okay. But then, yeah, we don't distinguish different type of use and type of nitrogen. Typically also at the level of the leaf. But that's part of, I think that to me, that's part of the attempt of the project to at the leaf level, a minima, to be more explicit and where to more explicitly locate or precise to which processes this nitrogen investment is done.